Okay. Our project is to genetically modify E. coli in cattle feed to reduce carbon dioxide production. To introduce our project, we plan on reducing the impact that cattle have on global warming. Cattle release large amounts of greenhouse gases into our environment, such as carbon dioxide and carbon monoxide. Our plan consists of reducing the amount of carbon dioxide emissions by genetically engineering the bacteria found in the rumen of cows. Reducing carbon dioxide emissions from cattle could potentially help to slow climate change and better our environment. Our solution involves modifying microbes to be placed in the rumen of cows. These microbes will help reduce carbon dioxide by using it as their sole carbon source. We will be using different genes from different bacteria and place them into the plasmid of E. coli. Through previous studies and shown in Figure 1, it has been confirmed that the addition of FDH, Rubisco, and PRK to the metabolic network of E. coli will induce autotrophic growth and therefore will lessen the amount of carbon dioxide produced by cattle.
The FDH gene comes from the organism Pseudomonas sp, which codes for the enzyme formate dehydrogenase, which catalyzes the oxidation of formate into CO2 and NADH. The PRK gene comes from the same organism, and it codes for the enzyme phosphoribulose. This enzyme catalyzes the phosphorylation of an intermediate of the Calvin cycle. The YSA08807 gene comes from P. putida, which codes for Rubisco, which is another enzyme that fixes atmospheric carbon dioxide. The last gene is the LuxCDABE operon from the P. luminescens organism. This operon codes for luciferase, which in the presence of its substrate flavin emits a blue-green light when active. This light emission allows for us to select recombinant bacteria without the use of antibiotics.
So we will be using polymerase chain reaction, or PCR, to amplify the number of copies of the gene. By using restriction enzymes, we will cut the LuxCDABE operon from the LuxCDABE plasmid. Then we would ligate these gene fragments to the open plasmid of E. coli using DNA ligase, and this will create our final plasmid. We would then insert the new plasmid into E. coli through transformation. We will plate this E. coli on an LP plate and check for bioluminescence. The flavin substrate will also be included to select for transformants. Transformants that emit a blue-green light mean that they are expressing the LuxCDAB operon. And these transformants, along with the flavin, will be added into the cattle feed. And this process is portrayed in Figure 2.
Okay, so upon doing this research for this experiment, we found some potential risks that could result. So one of them was that the genetically modified E. coli that we engineered could potentially overrun the wild-type populations that are found in the cow rumen. And it's also possible that the E. coli that's added to the feed could potentially infect the cows in other areas. The obvious benefit of the genetically modified E. coli is that it could reduce the carbon dioxide production by the cows. Although there's still a net production of carbon dioxide, it's ultimately less than what originally would be produced. There's also the fact that there's no known studies regarding the impact of increased carbon dioxide on the health of the cow and its functionality. So more research would need to be done on that. But overall, the project is feasible other than any difficulties that might occur from attempting to ligate genes from different or multiple different plasmids together. But other than that, the project was likely to be inexpensive and profitable. And it might require a large number of these modified cows to see a significant difference in carbon dioxide levels in the atmosphere.
So there are regulatory agencies that would oversee this project, one of which includes the Animal and Plant Health Inspection Service, and this would investigate the well-being of cattle. There is also the Environmental Protection Agency. This would regulate the safety of the genes and its impact on the environment. And lastly, the Food and Drug Administration would be supervising this genetically engineered bacteria and observe its effects on not only the cows, but also the consumers.